Gypb. Glycophorin B MNS blood group gene designation. Gypb, also known as sialoglycoprotein delta and SS active sialoglycoprotein, is a protein which in humans is encoded by the Gypb gene. Gypb has also recently been designated CD235 cluster of differentiation 235. Function. Glycophorin A, Gypb and B Gypb. This protein are major sialoglycoproteins of the human erythrocyte membrane which bear the antigenic determinants for the MN and SS blood groups respectively. In addition to the M or North and South or S antigens that commonly occur in all populations, about 40 related variant phenotypes have been identified. These variants include the Miltenberger Mi complex in several isoforms of Stoensten tau, also Dantu, SAT, Henshaw or MNS6, MG and deletion variants NA, SSU and MK. Most of these are the result of gene recombinations between Gypb and Gypb. Genomics. The gene is located on the long arm of chromosome 4 fork 28q31 and has 5 exons. It was first sequenced in 1987, the peptide sequence of 72 amino acids having been determined earlier that year. The gene has 97% sequence homology with the glycophorin A gene from the 5UTR approximately 1 kilobase upstream from the exon encoding the transmembrane regions to the portion of the coding sequence encoding the first 45 amino acids. There is a signal sequence of 19 amino acid residues. The leader peptide differs by 1 amino acid and the next 26 amino acids are identical. Amino acids 27-55 of glycophorin are absent from glycophorin B. This section includes an N-glycosylation site. Only O glycosylation sites are found on glycoprotein B and these are linked via serine or threonine. Residues 80-100 of glycophorin A and 51-71 of glycophorin B are very similar. The intervening residues in contrast differ significantly. The antigenic determinant for the blood group SS is located at residue 29 where South has a methionine and S a threonine. This is due to a mutation at nucleotide 143CT. The South antigen is also known as MNS3 and the S antigen as MNS4. It seems likely that this gene evolved by gene duplication and subsequent mutation of glycophorin at the transition site from homologous to non-homologous sequences can be localized within allele repeat sequences. Molecular biology. There are 80,000 copies of glycophorin B per erythrocyte. Both glycophorin A and B are expressed in the renal endothelium and epithelium. The first 40 amino acids of the mature protein are extracellular. The next 22 form a transmembrane segment and the remainder are intracellular. Blood groups. The MNS blood group was the second set of antigens discovered. M and North were identified in 1927 by Landsteiner and Levine. South and S were described later in 1947. The frequencies of these antigens are M, 78% Caucasian, 74% Negrid N, 72% Caucasian, 75% Negrid S, 55% Caucasian, 31% Negrid S, 89% Caucasian, 93% Negrid. Transfusion medicine. The M and North antigens differ 2 amino acid residues. The M allele has serine at position 1C at nucleotide 2 and glycine at position 5G at nucleotide 14, while the North allele has leucine at position 1T at nucleotide 2 and glutamate at position 5 at nucleotide 14. Glycophorin B carries the blood group antigens N, SS and U. Both glycophorin A and B bind the Vichigramini anti-N-lectin. South and S antigens are not affected by treatment with trypsin or sialidase but are destroyed or much depressed by treatment with papain, pronase or alpha-chymotrypsin. There are about 40 known variants in the MNS blood group system. These have arisen largely as a result of mutations within the 4KB region coding for the extracellular domain. These include the antigens MV, Dantu, Henshaw, Ors or Miltenberger rather than F4 and Stoensten A. Chimpanzees also have an MN blood antigen system. In chimpanzees M reacts strong but North only weakly. No mutants. Individuals who lack Gypb have the phenotype SSU. This may occur at frequencies of 20% in some African pygmies. In individuals who lack both glycophorin A and B, the phenotype has been designated MK. Dantu antigen. The Dantu antigen was described in 1984. The DAN2 antigen has an apparent molecular weight of 29 kilodaltons kDal and 99 amino acids. The first 39 amino acids of the DAN2 antigen are derived from glycophorin B and residues 40-99 are derived from glycophorin A. DAN2 is associated with very weak S antigen, a protease-resistant North antigen, and either very weak or no U antigen. There are at least 3 variants, ND, NE and PH. The DAN2 phenotype occurs with a frequency of DAN2 phenotype is ~0.5 in American blacks and 0.1 in Germans. Henshaw antigen. The Henshaw antigen is due to a mutation of the North terminal region. There are 3 differences in the first 3 amino acid residues. The usual form has tryptophan 1 serine 309 serine glycine 5, while Henshaw has leucine 1 serine 309 glutamate. This antigen is rare in Caucasians but occurs at a frequency of 2.1% in US and UK of African origin. It occurs at the rate of 7.0% in blacks in natal and 2.7% in West Africans. At least 3 variants of this antigen have been identified. Miltenberger subsystem. The Miltenberger Meyer subsystem, originally consisting of 5 phenotypes Meyer, VW, Mer, Hell and Hut, now has 11 recognized phenotypes numbered I to Roman 11. The antigen Mer is named after the patient. The original serum was isolated from a Mrs. Marl. The name originally given to this complex refers to the reaction erythrocytes gave to the standard Miltenberger antisera used to test them. The subclasses were based on additional reactions with other standard antisera. Meyer, W, Roman 7 and Roman 8 are carried on glycophorin A. Meyer is due to a mutation at amino acid 28 threonine to methionine CT at nucleotide 83, resulting in a loss of the glycosylation at the asparagine residue. Roman 2 is due to a mutation at amino acid 28 threonine to lysine C at nucleotide 83. Similar to the case of Meyer, this mutation results in a loss of the glycosylation of the asparagine residue. This alteration in glycosylation is detectable by the presence of a new 30-glycoprotein stainable with PHS. Roman 7 is due to a double mutation in glycophorin A, converting an arginine residue into a threonine residue and a tyrosine residue into a serine at the positions 49 and 52 respectively. The threonine-49 residue is glycosylated. This appears to be the origin of one of the Roman 7 specific antigens ANIC, which is known to lie between residues 40-61 of glycophorin and comprises sialic acid residues attached to oglycosidically linked oligosaccharides. This also explains the loss of a high-frequency antigen ENIC found in normal glycophorin A, which is located within the residues 46-56. Roman 8 is due to a mutation at amino acid residue 49 arginine threonine. Roman 8 shares the ANIC determinant with Roman 7. Roman 3, VI and X are due to rearrangements of glycophorin A and B in the order Gly-alpha Gly-B delta Gly-alpha. Roman 9 in contrast is a reverse alpha-delta alpha hybrid gene. VJ, Al, and Synta are due to unequal but homologous crossing over between alpha and delta glycophorin genes. The MyV and MyVJL genes are arranged in the same 5-alpha-delta-3 frame, where Synta gene is in a reciprocal 5-delta-alpha-3 configuration. Although uncommon in Caucasians 0.98% and Japanese 0.6%, the frequency of Roman 3 is exceptionally high in several Taiwanese Aboriginal tribes, up to 90%. In contrast, its frequency is 2-3% in Han Taiwanese Minnan. The Roman 3 phenotype occurs in 6.28% of Hong Kong Chinese. Roman 9 MS32 occurs with a frequency of 0.43% in Denmark. Stone's antigen. Stone Synta has been shown to be a product of a hybrid gene of which the 5-half is derived from the glycophorin B, whereas the 3-half is derived from the glycophorin A. Several isoforms are known. This antigen is now considered to be part of the Miltenberger complex. SAT antigen. A related antigen is SAT. This gene has 6 exons, of which exon I to exon Roman 4 are identical to the north allele of glycophorin A, whereas its 3 portion, including exon V and exon VI, are derived from the glycophorin B gene. The mature protein SAT protein contains 104 amino acid residues. Orisa antigen. Orisa appears to be a mutant of glycophorin A, but its precise nature has not yet been determined. Transfusion reactions. Both anti-S and anti-S have been implicated in transfusion reactions and hemolytic disease of the newborn. Anti-M, although occurring naturally, has rarely been implicated in transfusion reactions. Anti-N is not considered to cause transfusion reactions. Severe reactions have been reported with anti-U and anti-Miltenberger. Anti-MyIVW and MyRoman3 has been recognized as a cause of hemolytic disease of the newborn. Rasin has been associated with severe transfusion reactions. Other areas. Glycophorin B acts as a receptor for erythrocyte binding ligand EBM one of Plasmodium falciparum involved in malaria. Both the DAN2 and the SSU cells phenotypes have been shown to be protective against P. falciparum infection, while the Henshaw phenotype is not protective. Influenza and B bind to glycophorin B.